Everyone, welcome to a new lecture called as Quality by Design. Let's start with the definition. So, quality by design is defined as it is a systematic approach to the drug development that starts with predefined objectives and emphasizes on emphasizes product and process understanding and process control based on the principles of sound science and quality risk management. So, in the simple manner, how can we understand the definition of quality by design means? So, we are designing a process that can help us to achieve the good quality product. Quality by design has three main objectives. So, one is it focuses on the quality of the drug and its development, and the second one is risk management and risk assessment. Then the third one is continuous improvement and a journey towards the better quality products. So here process design mainly includes process understanding, product understanding, risk assessment and risk management. Okay, let us start with the one by one. So first we will start with the product understanding. So product understanding mainly includes API variability. API means active pharmaceutical ingredient variability, then excipient variability, process variability, packaging variability and other variabilities. So there are various analytical methods and powerful tools are there to evaluate the purity of the drug as well as the drug and excipient compatibility and drug interaction studies. So some are listed here. So among them, the first one is thermogravimetric analysis. So the principle of working of thermogravimetric analysis is based on the measuring of the changes that are taking place in the mass of a substance when it is continuously exposed to a elevated temperature. So when the sample is continuously exposed to the elevated temperature, so there are some changes which are taking place in the mass of that sample or the substance. So all these are studied in the thermogravimetric analysis. And the second one is called as DSC, that is differential scanning colorimetry. So it is a thermoanalytical technique in which the sample and the reference both are placed in a constant temperature. Then either the heat may get absorbed by the sample, which is called as endothermic, or heat may get emitted from the sample, that is called as exothermic. So then a difference in the amount of the heat energy required to increase the temperature of the sample and the reference is measured as a function of the temperature. So both the sample and the reference are placed or maintained in a constant temperature throughout the experiments. So here in this experiment, so the reference and the sample both are maintained or placed in the constant temperature, but the heat may get absorbed by the sample or it may get emitted from the sample, due to which there is some difference will be created in the temperatures. So that is analyzed, that is how much energy is required to maintain this balance between the sample and the then the next one is isothermal stress testing. So isothermal stress testing is a method used to study the drug and excipient interactions, or you can say the compatibility. And the next one is HPLC, that is called as high performance liquid chromatography. So used in the analysis of drugs and the pharmaceutical ingredients used in the separation, determination, identification, quantification of pharmaceutical ingredients, and used in the detection of many type of impurities in the drug formulations also. And the next one is X-ray diffraction. So X-ray diffraction is used in the characterization of the crystalline materials, measurement of the sample purity. So risk management is nothing but it is the systematic approach for risk identification, analysis of the risk, evaluation of the risk, reduction or removal of that risk factor. Now the advantages of risk assessment. So it prevents the production of defective product, helps in achieving the predefined desired quality product. So prevents the financial loss and helps in smooth running of the manufacturing process without causing any loss or disturbances. Then helps in achieving the desired quality product. The key elements of the QBD. The first one is define quality target product profile. The second one is identifying the quality attributes. The third one is determination of critical quality attributes. And the last one is critical process parameters. Quality target product profile. So it is nothing but a definite idea or a predefined summary or a aim or a purpose of the extent of the quality of the final product which is desired or required. So it is the basic and initial step in the QBD in which the formulator has a clear description about the type of the product which is required. So example, quality target product profile of a immediate release solid dosage forms for that includes tablet properties such or characteristics such as uh, tablet identifying and tablet identity and its appearance, assay and uniformity, extent of purity, extent of impurity, and dissolution rate. Critical quality attributes. So the critical quality attributes are the physical, chemical, biological or microbiological properties of the product. They should be present within the appropriate limit or in the particular range. Critical quality attributes play a very important role in obtaining the desired performance, quality, safety and as well as efficacy of the product. Quality attributes for drug and raw materials. Appearance, particle size, moisture, organic impurities, inorganic impurities, assay. Quality attributes for final product or dosage form or for formulation. So appearance, hardness, moisture content, content uniformity, dissolution rate, disintegration rate, microbial limits. Determination of critical process parameters. So it is defined as it is the measurable input or output of the process that can be controlled to obtain the desired product quality. Critical process parameters are mainly concerned with the process of manufacturing. Examples for critical process parameters. So the best examples for critical process parameters are here in the process of preparation of tablets. We prepare granules in the wet granulation method. By the wet granulation method, we prepare granules. So here in this process, the critical process parameters are the mixing time, the impeller speed, binding agent addition, and the temperature. And the impact of these factors, what we are going to get means that is called as potential quality attributes, or you can say the result of operating critical quality attributes. So here, due to these process, that is mixing time, impeller speed, binding agent addition and temperature, we will get the blending uniformity and the size of the granules, that is granular size, and the distribution of granules, that is granular distribution, and the moisture content of the granules. So all these results are the impacts which is obtained due to the critical process parameters. So the mixing time and the impeller speed will decide the blending uniformity and granular size, and as well as the binding agent decides the granular distribution, and the temperature decides the moisture content. And similarly, the next example is drying process. So in the drying process, the critical process parameters are drying time, that is the time of the duration of the time of required for the drying or used for the drying process, and type of equipment which is used in the drying process, and the input and output of the sample, that is the uh, in what way the sample or the sample which is going to subject in for the drying process is get entered into the equipment, that is the drying equipment, and how the resultant product is collected from that equipment, and the capacity of the drying chamber. Then another one is the results which is obtained due to these processes, uniformity in the moisture content. So the drying rate decides the moisture content of that product. The design space. The design space is nothing but the area where the process of manufacturing is carried out. So it is defined as the multi-dimensional combination of the interaction of input variables, example materials which are used in the manufacturing process, and the process parameters like drying, grinding, milling, coating, etc. So design space is nothing but the space where the manufacturing process is going to carried out, and in that place we are going to combine the Input variables means the raw materials and other things which are employed in the process of manufacturing, and the process parameters like the process which are used, that is like drying, milling, grinding, coating, etc. All those things which are used in the manufacturing process. So all these things will happen in the design space only, where the manufacturing process is going to carried out. So the manufacturing process is a complex dance between the various variables, or the that is elements, such as the materials being used in the manufacturing process, the and the technique which is employed, like drying, grinding, milling, coating, etc. and more. Each input variable and the process parameter, each of these combination play a very important role in shaping the final outcome. So the design space is constructed for a single unit operation or for multiple unit operations or for entire process. So design space is proposed by the applicant, or you can say the formulator or the manufacturer, in order to get the regulatory assessment and the approval. So the design space allow the space for the entire manufacturing process, starting from the raw material to the finished products. The design space includes machineries, and the proper ventilation should be there in the design space. And the next one is water supply, then power supply to run the machines, the safety measures, and the raw material store to store the raw materials, and finally the laboratories.